Hello, this is a video for those who've received their BeCrop Test results and would like a walkthrough of the report to better understand what those results mean. As a quick reminder, this is an analysis of the biological component of the soil. We can't see with this test nutrients themselves, we can't see pH, can't see texture. We're purely looking at the living fraction in soil, the organisms. What we've done, we've extracted the DNA from the bacteria and the fungi in the soil. We've snipped out just the piece of that DNA we need to identify who it came from. That gives us a very comprehensive view of who's living in your soil, and from there, this report will express to you, here's your soil's capacity to perform various agronomically relevant functions. Before we get into it, a quick note about scoring. You might well ask, okay, on my report, I see very low, low, medium, high, very high. How did you decide what was very low or what was high? On what basis? Are these just arbitrary values? We let nature decide. What we do is we have a database of many, many samples for various crop types. And so for that crop type, the 100, the top of very high, is going to be the highest functional capacity for a given metric or BeCrop index that we've observed in that crop. And the 0, the bottom of very low, is going to be the lowest score we've ever seen for that crop. And so where we place your sample is where it falls on that spectrum of globally observed instances. And that is true whether that function is normally distributed or non-normally distributed. If you're new to BeCrop analysis, I recommend that you skip page 1 to begin with. We will circle back to it at the end. Let's start with page 2. This is the health section. Health here does not refer to overall soil health, as in, say, like a Haney soil health test. This refers specifically to the level of pathogens, of disease-causing agents in the soil. So zooming in on this first piece, these are microorganisms that cause economically important diseases in the crop that you have assigned to your report. If the relative abundance of the diseases, the pathogen in your sample is on the high end of what we've observed over the last 11 years in testing soils of that crop, it will be highlighted at a high or very high risk level, and you'll see the detected organism listed. You'll also see here 27 out of 100 parcels analyzed are affected by charcoal rot. In other words, we see this pathogen to some extent in 27% of corn samples. We see Fusarium rot in 98% of corn samples. When looking at pathogen data, we always need to bear in mind the disease triangle. The presence and abundance of the disease-causing organism is only one factor in your plant getting sick. There also needs to be a favorable environment for that pathogen to infect the plant, to become virulent, and the plant needs to be weak enough, immunocompromised enough to get sick. So what we're reporting here in that first section is the pathogen piece. It helps to know if the enemy is at the gates. That does not itself translate to you're guaranteed to get this disease. It means we should be aware of what conditions lead to this disease infecting. When does it become virulent? When in the season? We need to be scouting this area, use this information to inform scouting or rotations or seed traits. This next section here will be pathogens that were detected in your sample, but they weren't, elevated compared to our global database of, in this case, corn samples. They weren't, starkly high risk, but we want to let you know that we see them because, of course, microorganisms reproduce. They can increase in number if the conditions are adequate to their needs, if the conditions are optimal for them. The last section here, you will see also, these are the pathogens that we did not detect. Keep in mind, we're a soil test. So any of these pathogens that are, say, translated by or communicated by equipment, by seed, by air through aerial spores, a soil test is not a surefire test for such pathogens. Think, for instance, powdery mildew. If we're finding it in the soil where it doesn't complete its life cycle, that's a sign that there's been enough powdery mildew in the air floating around that some got in the soil sample. But consider BeCrop Test a stronger screen for soil-borne pathogens than for airborne pathogens, tractor-borne pathogens. This last section here, biocontrol, these are the beneficial organisms that are armed and equipped to do battle with the pathogens. These are the secret service of our crops. They are able to defend our crop from the pathogen. Take a look at these two aggregates. Both of these were infected by Fusarium oxysporum, but only on the left did it become, well, did it run amok? Did it get out of control? That's because that aggregate on the left was sterilized. There was not life in that soil to prevent the Fusarium from getting out of hand. The soil on the right was alive. The Fusarium had a much harder time getting a foothold, and that's not even speaking necessarily of specifically biocontrol agents, which is what we're reporting here, such as trichoderma, which will infect a pathogenic fungi itself. It's the disease of the disease. It's the parasite of the parasite. It will eat Pythium, for instance, or Fusarium or Rhizoctonia from the inside out. Bactericide agents refers to microorganisms that produce antibacterial products. Insecticide agents are microorganisms, typically fungi, who suppress insects, often by infecting them and eating them from the inside out. And then finally, nematicide agents. This refers to microorganisms that will suppress, kill, antagonize, trap, decompose the eggs of, disrupt the life cycles of root-feeding nematodes. So when we say fungicide agents, we're not testing residues of fungicides. These are organisms. These are naturally occurring disease suppressors. So how do we put this together? You can look at your report and you can think, okay, I have X level of pressure from pathogen X. We saw a very high risk on this report for charcoal rot. So we got high abundance for charcoal rot. And we saw we had low abundance here of natural fungal pathogen suppressors, beneficial police force microbes. This would therefore, when it comes to charcoal rot, be a vulnerable soil. What we do about that depends on, have we already planted? Is there a time in the season that we can change up our rotations, select for charcoal rot resistance among our choice of seed, choice of crop? Is there time yet to inoculate our seeds with a beneficial charcoal rot suppressive organism? Do we need to do a rescue spray? These are some of the decisions that will be better informed when you know what your disease risks are and what your capacity to defend the crop is from that disease. Once again, I would like you to skip a page and go to the nutrient cycling section. I think it's wisest to start with the familiar things, such as disease-causing organisms. They're the living organisms in our system that most of us are most familiar with if we're new to testing soil biology. Let's go then to nutrient cyclers. Remember, this is a DNA test. We're looking at living organisms. We can't see the nutrients themselves. These are going to be assessments of the microorganisms that transform nutrients into and out of a form that your crops are able to take up and assimilate. So from here, let's look real quick. I'm not going to go into depth. We're not going to cover the whole nitrogen cycle. I just want to point out, in the nitrogen cycle, there are nutrient forms. We have nitrates, nitrites, ammonium. We've got atmospheric gaseous nitrogen into gas. There's also transformations. There's these white arrows. The vast majority of these white arrows are governed by living organisms. Everywhere you see a DNA symbol, microorganisms carry out that transformation, that step. So to take unavailable organic nitrogen and turn it into ammonium or nitrates that plant roots can take up, microorganisms have to be involved. Microorganisms are also involved in denitrifying, gassing off nitrates into nitrogen gas that leaves your system. That is fertility money flying away. They're also responsible for taking nitrogen in the air and fixing it into the soil, into a form that can then be transformed and assimilated by roots. Makes sense. There's also microorganisms that they all have their own needs for nitrogen. Every living organism needs nitrogen. And so there is a pathway where microbes are taking up nitrogen that might otherwise be available to a plant, and that's called immobilization. So let's look real quick. On your report, you'll see 6 nitrogen pathways. We have the supply lines you could think of as provision. You could think of as, they're not technically all mineralization, but you could think of them in terms of mineralization. We have our nitrogen fixers, like we said, that are taking into gas and are fixing it into ammonia and ammonium that could then be assimilated into proteins that could then be transformed into nitrates that are more readily available to plants. We have gain of nitrogen to ammonia. This refers to the decomposition of organic matter containing nitrogen, which would release inorganic nitrogen, ammonia, ammonium, that can then also be transformed into nitrate via the next metric, gain of nitrogen to nitrate. That's then the transformation from ammonia, ammonium into nitrite and then to nitrate, which is the dominant form taken up by plant roots. On the other side of the equation, we have the loss pathways, and I'm going to call them competition and loss, because the first one there, loss of nitrogen to gas, denitrification, those organisms, when there's lack of oxygen in the system, when the soil, say, is saturated for 24 hours straight, they will kick into gear and they will start transforming nitrate into gas, which comes off as a plume and leaves the system. That is a complete loss of nitrogen. Loss of urea by volatilization. This score it was created in order to be a great recommendation tool for nitrogen stabilization. The higher that value is, high and very high, means it is strongly recommended that you stabilize that nitrogen, because the soil has the workforce that can break down that urea quickly, which would spike the pH, causing some of that urea to volatilize as ammonia. These organisms don't directly cause the volatilization, but at high numbers, their activity will cause the pH to rise and lead to that outcome. And then finally, there's the loss of nitrogen to biomass. That's what I'm referring to as competition. That is not nitrogen leaving the system entirely. That is nitrogen taken into the cells of microorganisms, into biomass. That nitrogen is not leaving the system. So in fact, that can be a beneficial pathway, especially if you have sandy soils that are prone to leaching. Nitrogen that is held inside of the little fertilizer sac, which is a microbial cell, is safe from leaving the system. It's not currently available to the plant, but it can be remineralized later on. Meanwhile, it's not leaching, so that at least is positive. Bear in mind, plants can also directly consume microorganisms. Respects to James White, or I should say credit to James White for his research on this topic, but through a process called rhizophagy, that immobilized nitrogen can be directly taken in by plant roots consuming microorganisms, which is an essential part of root development and leads to the growth of root hairs. Okay. If you take, say, your supply lines and consider that inorganic nitrogen release, your competition and loss pathways, and consider that under this heading of consumption, because microbes are using that nitrogen for their own purposes, the ideal situation, I would argue, is to have high capacity to supply plant available nitrogen while also having ample immobilizers, that loss of nitrogen to biomass. I don't necessarily need to have high numbers of denitrifiers. I'd rather not, as that's a sign of conditions that are so low in oxygen that it's a favorable environment for denitrifiers to be pumping my nitrogen into the atmosphere. But immobilizers, organisms that break down urea, not necessarily a negative thing at all. And so I'd say that's balanced supply and high traffic. If I've got high capacity for release, low immobilization, I run a loss risk by having too much available nitrate beyond what the plant could assimilate. at any amount of time beyond what it needs. So if that's just floating around, it can be leached. I'd rather have microorganisms taking that up. On the other side, if I've got high immobilization and I don't have wide open supply lines, I don't have microorganisms that can supply the nitrogen I need, in that case, I face risk of a bottleneck. This is the most important quadrant for tissue testing, for prioritizing plant access. So in the yellow quadrant, I want to slow supply. I want a more gradual release. I want to foster microbial uptake of nitrogen, perhaps by supplying carbon into the system. Conversely, in the red, I want to provide nitrogen more rapidly. It might be inoculating with organisms that can transform nitrogen into plant available form. It could be the use of fulvic acids. It could be the way that I apply. I could split up my applications, for instance, wide drop or band it so it's more readily available to the crop. You will have, of course, you can pause if you want to go over this in more detail. I will cover the other nutrients with less detail because if you've wrapped your head around the nitrogen piece, you are in a good stead to be able to think critically and understand the others, which are a bit simpler. So phosphorus, without dwelling on this too long, a small fraction of it is available. Of fertilizer applied, on average 12.6% actually gets taken up by the crop. Most of it becomes fixed stably in inorganic phosphorus compounds. Some of it will be taken up by microorganisms, or is in crop residue, or is in manure, or is in microbial dead cells, and that's stable organic compounds. Microorganisms are responsible for re-releasing, supplying phosphorus across the season, whether by solubilizing unavailable inorganic phosphorus that's bound to the sand, silts, and clays that's complexed with irons and calciums and so on, or mineralizing unavailable organic phosphorus. So this first metric here, inorganic phosphorus solubilization, that is a blend of both mineralizers and solubilizers. This is, can your soil supply unavailable inorganic phosphorus into soluble phosphate? Can it supply unavailable organic phosphorus into soluble phosphate? This soil evidently has very high abundances of those crop provisioning microorganisms. Conversely, here we again have immobilization. This is microbial demand for the very same soluble phosphate that our plant needs to take up, are immobilizers. And so if you have an imbalance in favor of immobilization, then you risk a bottleneck. In this case, because phosphorus does not leach, it erodes, there's no concern at having very high solubilization and mineralization and very low immobilization. And I should mention the color and score over here on the left. That is the global score that is taking into account the balance between the supply lines and the competition and loss pathways. So phosphorus is coming up low because the three loss pathways amount to, an imbalance in favor of competition and loss as opposed to provision. The phosphorus is high because the solubilizers, the mineralizers, they outnumber the consumers. And then potassium, similar to phosphorus, but simpler even still because there is no organic form of potassium. There's only K plus ions. There's only mineral potassium. So it's just a matter of whether it is available in solution or whether it is, say, structural. It's part of the sand, silts, and clays, or it's non-exchangeable. It's secluded away in just cracks and crevices that are occluded by the soil itself. So microorganisms are responsible for solubilizing, breaking down into soluble form potassium that is part of the structural matrix of the soil. And that is what we have here, our potassium solubilizers, as well as potassium export pathways. There are specialized proteins that microorganisms have for pumping potassium ions out of the cell into soil solution, where they can be very conveniently picked up by plant roots. So those two pathways are encompassed by the potassium export score. And then once again, we have our immobilizers, microbes taking up potassium. And so your balance will be your global potassium score. And then finally, We offer indications of your soil's capacity to mobilize into a plant available form these 8 micronutrients. Is there anything more I should say about that? Probably there's a lot more I should say about that. In broad strokes, we would like to see a score between medium and high. Those are ideal scores. Those suggest that we have the capacity to mobilize these microorganisms into plant available form without mobilizing them so much that they could become phytotoxic. That's particularly relevant when it comes to, say, iron, copper, chlorine. Copper, for instance, it is sometimes, we have high numbers of organisms that can export copper from their cells, as in, get this out of here, I have too much copper, when copper is at levels that would become toxic to the plant. We sometimes see this where copper-based fungicides are heavily used. When it comes to having a low score in these, you can, say, circumvent the situation by applying, say, foliar, by applying chelated versions of these micronutrients to make sure that they're available to the plant. It's, of course, if you have multiple scores that are low, that's a very good reason to tissue test, to test sap, to see if these are actually preventing your crop from getting a hold of the nutrients it needs. These nutrients, if they're naturally very well exchangeable in the soil, this may not be preventing your crop from getting them. But if you're seeing deficiencies in a nutrient and you see low to very low scores here, biology may be very likely to blame. It may be cause for inoculation, use of organic acids, stimulation of the microbiome with carbon sources. As always, maximizing photosynthesis and improving soil structure by reducing disturbance. Those are always going to be the low-hanging fruit for improving soil function and soil health over time. Maximize photosynthesis and reduce disturbance, particularly tillage, where possible. I know I'm speaking in broad generalities. Contextualize everything to your operation, your crop, your needs. Now, before we leave our nutrient cycling page, we've got to cover carbon cyclers. We have first carbon fixers. These are organisms that can take carbon dioxide, gas, carbon dioxide from the air, and they can put it into organic matter. They can put it into sugars, into polysaccharides, into increasingly complex organic compounds. We have the organic compound degraders, which is to say decomposers. These are the guys that break down organic matter. They break down your crop residue, your stover. They break down wood chips. They break down manure. They're the ones who are going to break down organic matter and release the nutrients in it. Next, we have three metrics that talk about the oxygen preferences of the microorganisms. We have first aerobic respiration. These are the organisms that use oxygen in the process of respiring, in the process of producing energy. They must have oxygen in order to act, in order to do anything, in order to function. Then we have fermentation. This is looking at anaerobic respiration that does not involve the use of oxygen. Some of these organisms are able to deal with oxygen, able to tolerate oxygen. Others can't tolerate oxygen at all, but this is looking at the genes for deriving energy without oxygen. And then finally, we have the methanogens. This is a non-aerobic process. It doesn't happen in the presence of oxygen. And so you can probably realize before I say it, the combination of these three measures can give us a good sense of whether our soil is oxygenated, whether it is low in oxygen, whether it's aerated or poorly aerated. For instance, if you've got low numbers for aerobic respiration and you have high values for the organisms that can get by just fine without oxygen, that's a sign there might be poor aeration in your soil sample. That could be due to compaction, due to saturation, poor drainage, but it is definitely a cause to go out if you're not aware of an aeration issue and take out the penetrometer or pull out the shovel and see what's the structure like in the soil. Can this soil breathe? On the flip side, If you've got high numbers of oxygen breathing organisms and very few fermenters, very few methanogens, that might be a sign of, you probably want to see it in light soils, but a sign of potentially poor structure. You want to have some areas in your soil where oxygen does not diffuse in rapidly, whether due to aggregation or due to moisture, because oxygen doesn't diffuse nearly as well through water as it does through air. And so ideally, we would see substantial numbers of aerobic organisms and of fermenters. The methanogens we can do without, but as long as you do have oxygen breathing organisms, unless you're a rice crop, unless you're a crop that needs to be saturated, then that's the main thing. Do we have aerobic respirers, or are they completely outnumbered by the others? Okay, now we're going to backtrack a little bit, flip back, if you will, to the previous page, and we'll get into two very important sections that for many farmers, agronomists, and others who have not done any biological testing before, any DNA testing before, these will be new. The first group are plant hormone producing microorganisms. We have 6 metrics that touch on plant hormone pathways. Some of them are in a section on growth hormones. The other ones are stress-related functions. So first we have auxin producers, cytokinin producers, and gibberellin producers. These are all 3 plant growth hormones. They are plant growth regulators. Some of these are applied directly in a synthetic form in order to regulate stages in plant development. So here we have a quick look at the plant growth hormones. They are relevant to particular stages in plant life. For instance, gibberellic acid is a signal for seeds to germinate. Gibberellins, auxins, cytokinins are all extremely important throughout the life of the plant. I would say especially in early development when it comes to, we're looking at microorganisms in the soil that produce these plant growth hormones, but they're relevant throughout the life of the plant. Then ethylene and abscisic acid, we'll turn to those in a moment. But we're not looking at the plant's ability to produce these hormones. We're not looking at the presence of these hormones in the soil. We're looking at the presence of microorganisms that are able to produce plant growth hormones. Think about that. These are microbes that, because they have adapted to, developed to associate closely with plants, support plants, they're able to signal plants using the plant's own signaling pathways, signaling molecules. So consider, first of all, whether this is in the period of plant growth for your crop where these signals, these molecules are relevant. If it's out of season and I find I have a very low capacity to produce plant growth hormones, it's not particularly a high concern, because I wouldn't expect to see high numbers of these organisms if it's out of season, or if I just collected bulk soil for testing. If I didn't collect soil from the rhizosphere, where living roots are growing and exuding sugars and communicating with the microbiome, then neither would I expect to see large numbers of plant-associated microbes. But if I collected rhizosphere soil during the growing season, when there are developing plants, when there are roots growing in that soil, acquiring nutrients, a plant that's growing and developing, and I see low numbers of plant growth, of plant hormone producing microbes, that is alarm bells. That is the sign that your plant is having to put in all that much more effort on its own for lack of allies to support, guide its development, help it scavenge and locate nutrients, take off some of the metabolic load of producing these growth regulating compounds. And so your plant will not be able to thrive as well on its own than if it had its support staff, than if it had its team to back it up. And so this is a case where, what we do, the most effective way to foster plant growth promoting, plant allied microbes would be to use cover crops, to have living roots in the period leading up to when you're planting your cash crop. Improved soil conditions would help. You can directly inoculate plant hormone producing microbes directly onto the seed. For instance, There are many bacillus organisms, pseudomonas organisms that are commercialized and are known to carry the ability to produce these plant hormones. And of course, when it comes to synthetically applied hormones, if you're using a mixture of auxin or auxin cytokinin or those two plus gibberellin, the lower these scores, the more likely those products are to offer some benefit to you. The higher these scores, the more redundant those products are. Best case scenario, though, you have high numbers of plant growth promoting microorganisms. And now, without belaboring the point, we'll move on to this section, which is stress mitigating microbes, the capacity of your soil to help your plant adapt to particular stressors. There are 7 of these groups that we look at. Can I not make that slide go away? There we go. The first are, pardon the long word, exopolysaccharide producers. Exo means outside the cell, poly means it's a chain of, saccharide means sugars. This is just a chain of sugars that the microbes put outside the cell. What's the importance of that? They hold a lot of water. They produce kind of a protective zone that is soupy, that is sticky. You can see in this image, when you take images like this, you have to completely desiccate and dry down the sample. So the exopolysaccharides have been desiccated. They're completely dry. They've turned stringy. But these stringy bits, that's exopolysaccharides, extracellular polymeric substances. These retain water. They retain nutrients. These are the glues that build water-stable microaggregates. This is the foundation of soil structure. And so that will help with drought stressors. It will help retain nutrients, like I said, and it is going to be a soil building process. These are our architects in the soil. They produce rhizosheaths, more comfortable, more growth promoting environments around the root. Like I said, they produce water stable aggregates as well. These contribute to having a good result on your slake test. The next group are heavy metal tolerant plant growth promoting microbes. There are a whole host of different functions by which microbes do this. They take heavy metals out of circulation by precipitation. They sequester them away inside their cells so they're not available to plant roots. They can enable the plants themselves to become more tolerant by switching on their resistance pathways. Likewise, salt-tolerant growth-promoting microorganisms, analogous set of functions to the heavy metal microbes, but different specialization. So if you don't have salinity as a stressor on your operation, then this is not so relevant a metric. However, if salt is an issue on your operation, you would obviously want to see high numbers of organisms that can help your plants deal with that. You can have 2 plants in the same salty conditions. Those that have a supportive salt-tolerant microbiome will thrive much more, will tolerate the conditions much more than the one that lacks that support. And then likewise, think about the salt index on your fertilizers. If you tend to use high salt index fertilizers, then your plant will be able to tank the negative externalities of using that kind of fertilizer, the more of these microbes that it has available to support it. Siderophores, these are iron chelating molecules. So, which is to say, chelate is Greek for claw. They basically are able to strip iron out of compounds that it's complexed in where it's not plant available. And once they have chelated it, grabbed a hold of it from different sides, molecularly speaking, chemically speaking, it is then available for assimilation by the microbes or by the plant roots that intercept it. So if you have, say, alkaline conditions, or for whatever reason, iron is a limiting factor in your soil, it's not readily available, then this is a very relevant score to see improved, to see come up higher. Then the hormonal pathways on the stress side, you're probably aware of ethylene. Ethylene is a plant growth hormone that it's very important for fruit ripening, for instance, for transitioning from flowering to fruiting to maturing. It also, under stressful conditions, is produced as a stress signal. And if it reaches a certain threshold, the plant will shut down photosynthesis, shut down biomass production. it will shut down many functions and wait for better conditions. You don't want your crop to quit on you in the middle of the season. You don't want it to senesce early. And so what these organisms do, they produce a compound called ACC deaminase, which is to say this little molecule that could turn into ethylene. They pull an amine group off of it. They pull a molecular group off of it, and that prevents it from becoming ethylene. So same conditions, but the microbes are reducing the plant's stress level so that it's stressful out there, but the plant keeps on producing biomass, keeps on photosynthesizing, it keeps on synthesizing metabolites, and can basically tank the stress better than one that doesn't have the microorganisms there to soothe it, to help it cope, to say, look, we've got your back, keep on growing, we'll help you deal with the stressors. Salicylic acid producing microbes. This is the plant hormone that signals a systemic acquired resistance to pathogens that will parasitize plant cells but without killing them. You can think, for instance, of root knot or root gall nematodes. They don't kill the plant cells. They create themselves a little cozy nook and let the plant cells keep producing food for them where they can produce their babies. So salicylic acid is the signal to deal with those kinds of stressors, to turn on the plant's immune system. It also is relevant to dealing with a wide range of abiotic stressors, ranging from drought, heat, UV, waterlogging, both hot and cold. These microbes are helping the plant orchestrate its stress response. And then last group here, the organisms that produce abscisic acid. Abscisic acid is the the hormone that signals plants to shut their stomates when water becomes very scarce. So when you have microorganisms in the soil that can produce this, they're serving as an intelligence network about real-time changes in water availability. The plant that has a network of abscisic acid producers within its root zone, they're quicker to respond to changes in water availability. They're quickly to reduce losses of evapotranspiration through the leaves. And so they'll be more water efficient. If drought is a stressor on your operation, we'd like to see these organisms in higher numbers. We'd like to see, in fact, this and the last two in higher numbers. So brief example, these two plants are identical. Identical conditions. They both haven't been watered in these little pots for, I believe it was 5 days at the time this photograph was taken. There's only one difference between them. One of them was inoculated with a plant, with a stress mitigating plant beneficial microbe. It was a Pseudomonas species in this case. And so these microorganisms really can make a difference to whether your plant is able to tank stressors and keep on growing, or whether they wilt and shut down. So a framework for thinking about this, you know the stressors on your operation. We don't know what they are, but you do. So think about the stressors that your plants have to deal with, and then think about the microorganisms, the metrics of the 7 we just looked at, that are relevant to dealing with those stressors, whether they're heavy metals, whether they're drought, whether they are saturated conditions, flooding, whether they are, iron availability and alkaline conditions. And so if a stressor is not relevant to your operation, you don't care. That score is low on your test. But if a stressor is relevant to your operation, you absolutely care whether you have the microorganisms that can help your plants deal with it. And so if you have a distressed soil where that stress level is going to be high this season or is high right now, and you don't have the microorganisms to deal with it, in that case, there is strong reason, greater confidence for employing a stress mitigating product, a stress soothing product. There are many different kinds. I won't detail them here. Or inoculating with a microorganism that is able to help plants cope with that stressor. That is it for the stress mitigation section. Okay, home stretch, final section. We're circling back to page 1. Right up here in the upper right corner is a very important metric. This metric is called Biosustainability. If we have a soil health score, this is it. This metric is a single number that indicates basically how well integrated your soil is. So the lower this score, the more disjointed, the more competitive, the less cooperative, the more essentially dysbiotic, disordered your microbiome is. This this is not an absolute rule, but this is quite well correlated with the intensity of management, the level of disturbance that this soil has been through. Whereas on the other end of the spectrum, high scores, they reflect a microbiome that is integrated, that is exceptionally stable. The relationships within that community are well interconnected. It tends to be a more cooperative, more synergistic microbiome, and better reflects a natural microbial community. It is therefore a more typically durable, sustainable microbial community. And so, more information if you want to learn more about this, frankly, fascinating and really innovative metric, you can dig into this paper down here by Ortiz et al. In the middle of the page will tell you, hey, at a glance, here's your most important, or what we think are your low-hanging fruit for improvement. Not knowing anything else about the rest of your soil, you should always take your BeCrop results in tandem with other things you know about your soil, your in-field observations, infiltration and water holding, your nutrient levels. In the bottom left, you'll see total counts of bacteria and fungi per gram of soil in terms of the DNA recovered. And so if you're comparing two samples, suppose they're similar in terms of their functional abilities, but one has 1 times 10 to the 10th units per gram of bacteria, one has 1 times 10 to the 9th. Okay, the 1 times 10 to the 10th has more organisms, similar functional capacity, but a larger workforce. So they can do the same job and to the nth degree. It's very wise to have 2 samples to compare between each other, because if you've got the 1 sample, great, you've got it benchmarked against our global soils database. But it's even better if you've got 2 samples, one that has the problem you're trying to deal with, and the other one that doesn't have that problem, but is otherwise on your operation, similar climate, say it's a similar soil, make it as similar as can be without the issue you're trying to solve. That'll really help you narrow down specifically what are the biological factors that could potentially be driving this problem I'm observing. And then lastly, to the right, bottom right of this page, you'll also see fungal to bacterial ratio. Bear in mind, this is a ratio of the marker genes of the organisms. If you've heard about fungal to bacterial ratio before, that's usually a measure of biomass, of bulk. So be aware, fungi tend to have a lot more bulk in their cells. They're bigger cells on average. And so they'll have less DNA than the bacteria will in the sample. If you had a 1-to-1 biomass ratio of bacteria to fungi, you might have something more like a 1-to-50 or 1-to-100 in terms of DNA. If you requested any additional testing, Malic 3 analysis, Haney soil health testing from one of our partner labs, you'll find that data on the final page of the report. And then before we conclude, I want you to know there are some free tools available to you on your BeCrop account for delving deeper into these samples or making comparisons. For instance, there's a tool called the metric box plot tool in which you can load in 2 samples or 2 groups of samples, and this tool will automatically highlight the largest differences between those 2 samples. And if you click on one of those metrics, it'll give you a box plot if there's multiple samples in each cluster to show what the variance is, what's the spread of results between these 2 groups. I use that tool just about every day. There is another tool for comparing the relative abundance of particular microorganisms in the sample, which is useful in specific circumstances where the customer, the tester, is interested in particular microorganisms, be they beneficial or pathogenic. There is the heat map tool, which suppose you want to compare samples, but you don't want to average them, throw them in a cluster. The heat map tool lets you lay many samples side by side, each as an individual column or row. You can flip it so that you can look for differences or patterns between them across fields or years or seasons. And it's a lot easier than flipping back and forth between multiple PDF reports. There's a tool for exploring all of the organisms that were detected in an individual sample. And then finally, there's a tool, this one is called the Explorer tool. And then this one is the co-occurrence tool. It lets you pick 3 samples or 3 groups of samples, and it will automatically tell you how much overlap is there between them in terms of which organisms live in each sample. And you can get a list of, okay, these organisms are completely unique to this cluster of samples from these fields, or these microorganisms are unique to my high ground, my lowlands by the river bottom, so on and so forth. You can find guides for how to use each of those free tools at the BeCrop Help Center. I hope you're feeling much more confident about your ability to grasp and understand and interpret your BeCrop report. And if you have any breakthroughs, being able to glimpse now into the black box of the soil microbiome, if you solve problems that have been intractable for years, or if you have further questions or puzzles you'd like help to unpack, please feel free to reach out through the website. We like your success stories and to continue to help in areas that need additional code cracking. There are lots of resources in the BeCrop Help Center, and there's also a good many resources on YouTube and on our website in the form of webinars. So if you want to dive into the rabbit hole and really develop your expertise in understanding and managing soil biology, there are the resources available, but don't overthink managing biology. All this information can be overwhelming. Find an area you want to focus on, do some experimenting, try out some interventions for that, test to see how it's working out, observe. But most importantly, remember, don't overthink biology. Maximizing photosynthesis, minimizing disturbance are some of the most important things you can do to give your microbiome a chance to work for you. Give your soil workforce an opportunity to work for you and not against you to support your crop. And I think you'll find that your soils function better, are more resilient, handle water better, handle nutrients better as you start to build the living fraction in your soil. Thank you very much. It's been a privilege, and all the best to you and yours and your work.